Hello everyone, I am Dr. A. Muriyasan, Associate Professor from EEE Department in KSR Institute for Engineering and Technology. In this session, we discuss about components of optical instruments, especially spectroscopy. Spectroscopy is one of the optical instrument. Any type of spectroscopy have these five major components. The first and foremost component is the light source. Normally a spectroscopy is used to analyze any substances or sample how the sample will be reacted with the light. So that will be the role of spectroscopy. For that we need the light source. So the first and foremost component is the light source, which will give the light signal to the sample. So from the light source it will give various light having the different wavelengths. So for that analyzing the sample we need particular wavelength of the signal. For that we need a wavelength selector. This wavelength selector is used to select the particular wavelength of the light signal. So after that filtering the particular wavelength signal, that signal will be given to the sample. This sample may react with the given light signal. Either the light may be absorbed or emitted or scattered. After the emission or scattered or absorption, the light signal goes to the detector. Detector is a transducer. What is a transducer? Transducer will convert the physical quantity into electrical quantity. Here the detector will detect the signal from sample, light signal from sample to the electrical signal. So you can use any photo detectors which convert the light signal into electrical signal, like a photodiode, photo transistors, photo multiplier tube. You can use any one of photo detector which converts the light into electrical signal. Once it will be converted into electrical signal, the signal is related to the sample output. This electrical signal will be given to any readout instrument or processor. Before going to any instrument, if you need any signal processing means you have to amplify the signal. Suppose if there is any low value from the detector, you need to amplify. Suppose if there is any noises in electrical signal, you have to filter. So that's why we need some signal processing. After the signal processing, it will be given to readout or any processes for further processing. So this is the overall setup for any optical instrument or a spectroscopy. Already said, it have first handling the stable source which produce the EMR, electromagnetic radiation, or it will be act as a source of radiation. So in the next you have to device that is called wavelength selector. We may use monochromator or grating to select the particular wavelength of the signal. Next you have the transparent container which holds the sample. Why we are having the transparent container? The transparent container which allows the light passes through the sample. So that's why we are using transparent container. The next after that, after the sample you have to use some detector which detects the light signal from sample. Then it converts the light signal into electrical signal by using any photo detectors. Once if the get the electrical signal, if the amplitude is very low, you need you do you need to amplify. So you need amplification. If there is any noise, you have to need filter. So we need some signal processor to process the electrical signal. After the signal processor, it will be given to readout which displays the transduced signal. So these are five major components present in optical instrument, especially spectroscopy. So here these are the six different methods available in spectroscopic method. So first type will be the absorption method. Absorption method means the sample which is going to be analyzed may absorb the light. Which are the sample which going to absorb the light? That type of spectroscopy called as absorption spectroscopy. The next will be the fluorescence and phosphorescence, the material which may light or scatter the light, which will be emit the light or scatter the light. That is the property called fluorescence and phosphorescence. Whenever substances, when you are passing the light to the substances may produce different light signals. That is the property called fluorescence and phosphorescence and scattering. So these 2, 3 and 4, fluorescence, phosphorescence and scattering, is the property. Whenever the sample receives any lights, it may produce some different light signals. That's why that's a proper, that type of sample we can use fluorescence spectroscopy, phosphorus spectroscopy and scattering spectroscopy. Then the final one will be the emission and chemulence. So emission and chemulence used for, if the sample, it may produce the light without any light source, the sample itself produce the light, that due to the chemical property of the sample it may produce by the light signal. It's by own, by using the chemical reactions, that will be comes under the emission and chemulence. So depending upon the scope of these methods, the arrangement of the instruments may, the arrangement of the components may change. For example, so here we go for absorption is the normal method. You have a light source. From the light source you have to select a particular wavelength. From that particular wavelength, a particular wave light signal will be selected from the light source. Then it will be passes through the sample. The sample may absorb some of the light depending upon the property. Then the remaining emitted light, transmitted light will be detected by the any photo detector, which converts the light into electrical signal. Then the electrical signal will be given to signal passing. Then it finally gives us the output. This is the absorption method. So next, we have to, I already said, we have to measure the fluorescent, phosphorus and scattering measurement. In that said, we have a source. Then from the source you have to select a particular wavelength from the wavelength selector. Once the light will be given to the sample will produce many different light signals. Due to the property of fluorescence, phosphorus and scattering of the property of the sample, it may produce different light signals. So you need another wavelength selector to select the particular wavelength from the sample. So here we need one more wavelength selector to select the particular wavelength from the sample. Then the wavelength, after the selection of the wavelength signal, it will be detected by the photo detector, which converts the light signal to the electrical signal. Then it will be given to the display. So in this method we need two wavelength selector. One is used for input side, the another one will be used in output side. Then the third part will be the emission and chemulence property. I already told that the sample itself produce the light due to the chemical property or chemical reaction. The sample itself produce the light. So here there will be no need for separate light sources. So you have the sample itself produce the lights. The sample, it will be act as a light source. So the light signal, the light from the sample will be given to the wavelength selector. The wavelength selector is to select a particular wavelength from the sample, that the wavelength and the particular signal will be detected by the photo detector. The photo detector will give the electrical signal to the signal passing and readout. So here there will be no separate light source for the this method. Here the sample will be act as a light source. So these are different spectroscopic methods available for analyzing the different types of samples. Thank you.